Welcome to this webinar. This is the second one in a series of 3 webinars on microfluidics. My name is José Alves and I'm your host today. Dr. Monica Brivio will start her webinar about using microfluidic technologies for point-of-care diagnostics in a minute. Monica is Development Manager at Micronit. During this webinar, you can ask questions through the chat. A selection of your questions will be answered live at the end of the webinar. All questions, however, will be answered and sent to all attendees by email. We will anonymize the questions first. The webinar will take about 60 minutes, including the Q&A about 5 minutes. Now let's go to Monica and enjoy this webinar.
Thank you, José, and welcome all of you to this webinar, which today will focus on the use of microfluidics for in vitro diagnostics applications, and in particular for at the point of care settings. My name is Monica. I've been working here at Micronit for almost 8 years now, and for about almost 20 years, so since I started my PhD, I have been working in the field of microfluidics, always at the interface between microfluidic technology and application technology. So I've seen and I've done a lot of assay transfer from the lab onto a microfluidic format. My goal for today's webinar is to show you how microfluidics are key enabling technologies for the development of diagnostic products, but I hope I will also be able to show you how microfluidic technologies are driving innovation and growth in the diagnostic market, in particular by bringing diagnostics closer to the patients. And by closer to the patient, I mean both in terms of point of care, but also in terms of personalized medicine and diagnostics. These are key concepts that will come back a few times during the webinar today. Now that you know who is going to talk to you from behind the scenes, let's switch to the presentation, and I will be happy to guide you through the slides. So I hope you will enjoy this webinar. Okay, let's start. So I'm going to start with an introduction about Micronit. First, I will tell you who we are, what we do, and how we work with which materials and which technologies. Then I will focus on the in vitro diagnostic market, and in particular, I will highlight some needs and trends we see in this market. And then I will make a link to enabling technologies, namely microfluidics and microtechnologies more in general. As José already mentioned, at the end of this webinar, there will hopefully be enough time to answer a few questions. So please type in all your questions as they pop up throughout the whole presentation, and we will collect them and try to answer a few of them at least at the end of this webinar. Let's start about Micronit. At Micronit, we believe in a future where people age healthily. This is a very straightforward vision statement, which I'm sure many of us recognize themselves in. But how do we at Micronit strive to fulfill this vision? Well, Micronit is the leading innovator and global partner in design, development, and manufacturing of customer-specific product based on microfluidics and MEMS technologies, mostly focusing on healthcare applications. So primarily, we are an ISO certified manufacturer of microfluidics and MEMS-based consumables, but we also have over 20 years of experience in designing and developing microfluidics and MEMS-based products. So this makes us a suitable partner not only for manufacturing, but also to help our customers throughout the whole value chain of their products, all the way from idea generation to manufacturing, going through the steps of development, like feasibility, proof of concept, and the whole scale up. Based on these capabilities, we have been for 20 years and keep on serving key players in the life science, diagnostics, and biotechnology market. Our team counts more than 100 people based in 2 different locations. Our headquarters in the Netherlands, Enschede, from where I'm giving this webinar, and Dortmund, Germany, which mainly focuses on MEMS technologies. And I would also like to mention that we have quite a strong core technology team, which takes care of innovation. So their main goal is to extend and consolidate our toolbox, technology toolbox. And this is very important for a company like Micronit, the ability of innovating, to be able to help our customer to be successful in their markets. Speaking of markets, I already mentioned that we serve mainly applications within the healthcare market. And there are a few market segments that we particularly focus on that are represented, or they're shown in this slide. As you can see, it's quite a nice mix of well-established markets that are established but still dynamically growing, mostly enabled by microfluidics, and a few emerging markets such as liquid biopsy, single-cell analysis, and cell culturing. All of these markets are enabled by microfluidics and microtechnologies in general, I would say, and most of the markets shown in this slide, on this slide, except for cell culturing, they also partly share some of their workflow. Just to give an example, liquid biopsy relies on next generation sequencing workflows for the downstream analysis. And it also relies on single cell analysis workflows for sample preparation, in particular for CTCs. So all these markets are, as I said, enabled by microfluidics and microtechnologies. And all these markets, although one more or less than the other, are characterized by a number of trends that we have observed already for a few years. In particular, I would say that the main trends can be summarized into 3 ones: high throughput, high contact, and automation of the workflow. As an example, so high throughput means basically producing a lot of data in one test. And to give you an example of this, I would like to mention digital microfluidics, so droplet generators, which are microfluidic systems that can produce droplets, and these droplets offer a microenvironment where all assay can take place. So an assay for analysis of a single cell, or even of a single molecule, can be done inside a single droplet, and this can dramatically be parallelized, thereby increasing the throughput. On the other hand, high content, an example of high content means collecting a lot of information about one entity. So, for example, looking at cells, instead of analyzing a population of cells and collecting an average signal, which provides average information about the cell population, one can focus on one single cell, isolate it, and then analyze it for its genomic, proteomic, metabolomic content. The last trend that I want to mention is the automation of the workflow. This is a trend that we observe in most of the markets we're active in, but in particular it's relevant for in vitro diagnostics, and in particular at the point of care settings. In point of care settings, the ease of use of a test is crucial, because you don't always have a trained personnel. Actually, most of the cases, the test is run by a non-trained personnel. So the complexity has to be moved from the user to the consumable, to the test. And this can be done by automating the workflow, which in turn can be done by adding functionality to a cartridge. So in terms of technologies, this translate into a high level of integration of functions, which is done by bringing together technologies like microfluidics, MEMS, micro-optics, microelectronics, to create systems or solutions with a higher value. And this is also something that we have recognized at Micronit already for a long time. And after creating a very solid basis of microfluidics, MEMS, and microfabrication technologies, for a few years now, we are developing capabilities that allow us to bring all these technologies and together to enable automation of the workflow. What does this mean for our customers? For our customer, this means that we can support in the development of microfluidics-based products in all materials or groups of materials that are typically used in microfluidics. Think of glass, silicon, different kind of polymers, but also, very importantly, we can offer hybrid solutions, hybrids of different materials. This is, again, very key for point-of-care testing because, again, having to integrate many functionalities on the same cartridge, the same cartridge will have many different requirements, functional requirements, but also economical requirements, user requirements, and it can be challenging to be able to meet all requirements by using just one material. So this is where hybrid integration of different materials is key. Of course, if we are able to supply products based on all these different materials, we need to have also a toolbox of technologies that allow us to structure different materials and create these systems with a higher value. So, for example, in this slide, on this slide, you see, which is not comprehensive, you see a few examples of technologies that we have in our toolbox. For example, for structuring glass and silicon, we use lithography and etching, wet and dry etching. For polymers, we use milling, embossing, and injection molding. And of course, once these structures are fabricated, we also need to seal them to be able to run fluidic workflows in them. And we also need to have a pool of capabilities, general and back-end capabilities that would allow us to add more functionalities. For example, adding reagents, dry reagents or liquid reagents, again, adding hybrid components, hybrid elements, for example, sensors, and so on and so forth. I will get back to some of these technologies, at least the ones relevant for in vitro diagnostics, later in the presentation. So beside extending capabilities within our in-house toolbox, we also recognize the importance to be able to offer more value to our customer of working with an ecosystem of selected partners, of strategic partners. As an example, I would like to mention the Flow Alliance, House of Microfluidics, which was established 3 to 4 years ago between 3 companies, Micronit, Axicon, and Elfood. And Elfood has 8 years of experience in high volume injection molding and in assembly, automated assembling of precision product. And Axicon has experience in high quality mold making and in the development of the molding process. So in this way, we can offer to the market a broader set of capabilities that allow us to meet all customer requirements for consumables for different applications, but in particular for in vitro diagnostics. I would like now to focus on the in vitro diagnostic market, make a link to the point of care settings, and then a link to the technology, see how microfluidics can enable development in this market. So first of all, I would like to start with some definitions and also to mention what the value of in vitro diagnostics is. In vitro diagnostic tests are tests that are done on samples such as blood, saliva, urine, or other tissues that have been taken from the human body. In vitro diagnostics are extremely valuable resources for disease prevention, detection, and management. And to appreciate their value, one should think that in vitro diagnostics influence more than 2/3 of clinical decision making, while accounting only for just a couple of percentage of the global healthcare spending. So they are an extremely valuable tool in healthcare. And in modern healthcare, in vitro diagnostics goes far beyond than just providing a diagnosis to a doctor, saying whether a patient has a certain disease or not, and what disease does a patient have. It also provides critical information at every step of the patient pathway. This is represented here in this image, from screening of the population to provide information on the predisposition of the population to get a certain disease, to prognosis, to predict the outcome of a specific disease, stratification, to make decisions, to help making decisions on what therapy for what group of patients, and finally for monitoring the outcome of the therapy. So basically, in vitro diagnostics is changing the way that healthcare is delivered to the public. And we see two main trends in vitro diagnostics that are personalization and decentralization. So personalized medicine focuses on giving the right treatment to the right patient or group of patients at the right time. In traditional medicine, the same therapy is given to the whole population, and as a result, a few people will benefit from the therapy, a few people will not see any effect, and a few people will even experience adverse effects. So with personalized medicine, what happens is that different therapies are given to different groups of patients based on their genetic makeup, genetic and biological makeup, to be able to maximize positive effect of all therapies. And how does diagnostic come into this picture? Well, diagnostics, which for this application is called companion diagnostics, has a role in enabling stratification of patients. Diagnostic tests can be used to basically define the genetic and the biological makeup of patients so that patient can be stratified into different groups and given the right medicines. The second trend of in vitro diagnostics is decentralization of healthcare. What we see is that in vitro diagnostic tests are moving from centralized laboratories to different point of care. Point of care can mean anything from near the patient bed in hospital or in a small hospital lab next door to the patient bed, or to the general practitioner, and all the way to the home of the patient. To appreciate the different flavor of point of care testing, I have collected here a few examples of commercially available point of care tests, which are used in different point of care. All of them have a common feature, which rely basically on microfluidics. So they all work with microfluidics, and they go from handheld systems such as the Abbott iStat, which is a reference for patient bedside testing in hospitals. It's used for blood chemistry, cardiac biomarkers, so it runs some blood chemistry and some immuno test. It provides results in a couple of minutes and is used in 10,000 healthcare facilities globally, so it's very much used. But we also have systems such as the Gene Expert, which is also used at different point of care in the hospital, but also at the general practitioner in the express version, and it relies on molecular diagnostics. Another example that I find interesting to mention with this point of care in vitro diagnostic test is the MySeq diagnostics by Illumina. This system is the first FDA-regulated CE IVD marked NGS platform, which is used for in vitro diagnostic testing, and it relies on next-gen sequencing workflows, which typically we see in centralized laboratories and used for life science research applications. So again, once more in this slide, the main point is that point of care can mean many different things, and as such can mean many different requirements. So this diversity in type of in vitro diagnostic test also means that the relative level of needs for different type of in vitro diagnostic test may vary depending on the type of test, on the point of use, on the user. However, there are a few key customer needs that need to be met for an in vitro diagnostic test to be successful. And they are automation, which goes hand in hand with ease of use. That's what we also already saw a few slides ago. Platform performance. A new test needs to perform equally good or better than existing ones. Turnaround time, typically point of care tests need to be fast. And instrument consolidation, that's the holy grail of in vitro diagnostics, having one instrument which can run all sorts of different tests in cartridges that at least share the same footprint. Now, if we read these key customer needs next to the key enabling features of microfluidics, it becomes clear why microfluidics and microtechnologies has already been used for in the field of in vitro diagnostics for innovation. By miniaturizing and integrating functionalities and components, microfluidics enable automation. And they also offer a micro environment where an assay can be carried out with a high level of control. So let's now focus on microfluidic-based point of care tests. And I would like to start with an example to which, everybody can relate to, because it's about the current COVID-19 pandemic. On this slide, which was kindly shared by Yole Development, we can see a number of molecular tests based on microfluidics that are at different stages of development, targeting the SARS-CoV-2, which is the agent causing COVID-19 infection. And this snapshot refers to 1.5 months ago. So I'm pretty sure that I'm already quite impressed by the number of microfluidic-based molecular diagnostic tests for point of care testing of the COVID-19 infectious agent. And I'm pretty sure that if now 1.5 months later we're going to take another snapshot, we will see that probably this graph gets even more populated. So the COVID pandemic has highlighted the need for rapid, actionable results at the point of care, which is not only true, in fact, for this target for the COVID-19, but it's true for an increasing number of test panels. Now, it seems that molecular diagnostics, molecular tests are key to obtain these rapid, actionable results at the point of care. But this category of tests relies on workflows that are quite challenging to be transferred to microfluidic platforms. At least it used to be like that. However, due to the advances done in technology recently, and in particular the ability of combining multiple functionalities on a single cartridge to enable automation, it's this type of tests are making it to the market, and this is what we see also in this graph. Now, if we look a bit closer to the microfluidic-based point of need testing market, we see that the green part, molecular diagnostics, is already accounting for more than half of the market value today, and it's expected to grow to 70% of the market value in 2025. with the market value expected to be in 2025 to reach $10 billion. Now, but let's see how such a molecular diagnostic workflow look like, because this is in the end what needs to be transferred onto an integrated cartridge to allow automation of the workflow at the point of care. A molecular test typically require a number of steps, starting from sampling and sample loading. The sample could be a body fluid like blood or saliva, or a swab. Cells need to be extracted from the sample and need to be, we need to perform a workup step. Workup can mean different things depending on the type of sample. Typically, cells are lysed to extract DNA material. In the case of virus, for example, for the SARS-CoV-2, RNA is extracted from the virus. Then RNA can be reverse transcribed into DNA, and the next step would be a DNA amplification. Amplification which allows making many copies of the target DNA to be detected. Now, the next step would be a detection step, which can be done in real time, but real-time PCR when fluorophores, specific fluorophores are added to the reaction mixture, or it can be done as an endpoint type of detection when the amplicons or products of the PCR are hybridized onto a microarray. What's important to notice here is that such a workflow requires a number of fluidic steps, but it also requires some temperature control. In particular for PCR, we need to have thermocycling, with fast thermocycling between 2 or 3 different temperature levels, going up to 94, 95 degrees. It requires integration of a detection or a readout system. So a lot of functionalities need to be integrated on a cartridge to be able to perform such a workflow. And the same counts also for another type of test, which is very relevant for point-of-care testing, which is immunoassay. While molecular assay have nucleic acids as targets, so DNA and RNA as target molecules, immunoassay are used for the detection of protein biomarkers. They are also used a lot in point of care, and they also require a number of fluidic steps, starting from sampling. The analyte needs to be captured or extracted and purified, which is done in this case by antibodies that are typically functionalized on the microfluidic channel. And additional reagents need to be brought in to provide labels that in the next step, when an additional fluidic or an additional reagent is added, will provide a signal that can be detected. A couple of other fluidic steps might be needed throughout this workflow for washing to provide enough sensitivity and specificity of this detection. So in all cases, for both immunoassays and molecular assays, we see many steps, fluidic steps, a lot of functionality on a cartridge. And the cartridge needs to be used at the point of care. So the cartridge we have already established need to be simple or easy to use. So the complexity is being moved from the user to the cartridge, which means that all the functionalities are integrated on a sample to answer type of device. Typical functions that need to be integrated on such a cartridge start with sample preparation, flow control. We have seen that we have a number of typically sequential fluidic steps to run the whole workflow. We might need to integrate reagents, either dried or liquid, and to coat the surface to provide specific properties to the surface, for example, anti-fouling. And finally, we might need to integrate hybrid elements, for example, sensors. In the next 4 slides, I'm going to focus on each slide on one of these functions, and I'm going to show you a few examples using demonstrators that have been developed within our core technology group mostly. And these technologies can be customized in customer products. Let's start with sample preparation. So there are different strategies for sample preparation. That depends on the type of test. It depends on the type of sample. It depends on the on many factors. But in many cases, or typically, a filtration step is required. It can be required, for example, for removing particles from the sample that otherwise would clog the channels. But it could also have a function of separating different components in a sample. For example, starting from whole blood, we might want to perform an analysis on biomarkers that are present in plasma. The filter separates the cellular component of the blood from the plasma component where the test is going to be carried out. These kind of filters, depending on the sample and depending on product requirements, they are commercially available, and what we do, we can integrate them. We can design microfluidic-based systems that can accommodate, integrate these filters. But we can go one step farther and also make some sort of smart type of filters, like in this demonstrator, which was developed in collaboration with the NEST and CNR from PISA in this SmartTool project. Here we see a very simple device with an inlet where blood can be loaded, a straight channel, and an outlet. If we look inside the device, inside the channel, we see that the channel presents 2 compartments, and the compartments are basically formed by an interposed membrane. I believe that in this case the membrane was in PET. So the membrane has a double function. It functions as a mechanical membrane for filtration. So when a sample of blood, all blood, is inserted in the channel, only the plasma with the soluble plasma biomarkers will move to the bottom compartment of the channel, which might be functionalized with antibodies against these biomarkers, these protein biomarkers. So we can carry out an immunoassay at the bottom of this channel. What happens at the top of the membrane, if the membrane is functionalized with antibodies that are against specific cells in the blood sample, for example, like in this case, monocytes, then the monocytes will bind to the antibodies on the surface of the membrane, whereas all other blood components, red blood cells, for example, will be moved towards the waste. So this is, let's say, a smarter membrane than just a membrane for filtration, which also has some active component. Another functionality that might be useful for running assays is volume metering. Metering volume is quite important for quantitative tests because we need to know how much of the different components we have in such a test. And there are different ways of metering volume, but the one that I've selected to illustrate it is based on the geometry of a channel of a reservoir, which is made on chip and which is loaded by capillarity, and valves placed in specific positions of the reservoir will prevent the liquid from leaving the reservoir until a specific trigger occurs. Bits-based assays are quite commonly used for all sorts of different assays, immuno or molecular assays, for as a solid support. They provide quite a standardized surface and a large, because they are produced in batches, in large batches, and they can also, and they provide a large surface to volume ratio. So they can be used to capture target molecules in samples, being those antibodies or proteins or nucleic acids, and they can manipulate it with, in different ways to be able to retain the analyte of interest during washing of the sample. In these two examples that I show here, we have different kind of beads that are handled in microfluidics using different approaches. This first example uses polystyrene beads that are coated with antibodies against ELISA products. They were used for an ELISA, beads-based ELISA, and they were captured inside this channel. The channel in this case was a glass channel, 700 or 800 micrometers wide and only 8 micrometers deep. And a filter, a sort of mechanical barrier, was fabricated inside this channel, leaving only 1.5 micrometer gap, so that the beads could not escape and would be trapped, while the liquid, the sample would leave the, without the analyte, of course, would leave the microfluidic chamber. And in this case, this chamber is, the beads are facing a sensor. I will get back to that in another slide. Another example of beads-based assay is offered by this demonstrator, which was developed within the CardiaTool project, where planar coil is used to pre-concentrate magnetic nanoparticles that are used to capture an analyte that will be then measured in the next portion of the device. I will get back to that also in the hybrid integration session of this presentation. Methods to sort cells are also often used in sample preparation protocols when cells need to be sorted and captured for further processing. My colleague Mark Olderikering 2 weeks ago in the first webinar of this Micronit webinar series already extensively explained this technology. So here I'm just going to mention a few, just to mention the technologies. And so, for example, we can have acoustophoretic separation, which is enabled by acoustic waves, basically, that act on the particles or cells that need to be separated and concentrated. In this case, we have dielectrophoresis. Separation is based on electrical properties of the particles or cells, and arrays of interdigitated electrodes are used to apply the actuation here and separate cells or concentrate cells and particles. In pinch flow fractionation, a combination of geometry of the channel and flow properties is used for separation purposes. And in the last example, deterministic lateral flow, lateral displacement, which is also known as DLD, is used as shown in this animation as a sort of coin sorter to separate cells based on their size. And as our customer GPB does, as you can see in this movie, a long channel featuring this coin sorter is used to separate red from blood cells, from white blood cells, which can be seen by the different intensity of the red and the white, milk white colors in the channels. The second functionality I want to talk about is flow control. There are different kinds of flow control to be integrated in microfluidics, but definitely flow control is a key functionality because all these workflows require a number of steps, a number of addition of reagents, a number of washing steps. So this is really a key functionality. There are different strategies for this, and today I'm going to show two of them: active flow control in this slide, and then capillary flow control in the next slide. So active flow control is known for a very long time, at least these kind of valves, membrane valves or peristaltic valves. They are based on two rigid substrates that are microstructured with a fluidic layer and a pneumatic layer in case of pneumatic actuation, and a flexible layer sandwiched in between the two rigid layers. There are different configurations, normally closed or normally open, and you see up here. I'll just take the normally closed to show what happens. So in a normally closed valve at rest, the channel is closed, so there is no flow, and when actuating the membrane, for example by applying vacuum, thereby lifting the membrane, the channel opens up and the flow can continue. In a normally open valve, actuations happen the other way around. So at rest, when the valve is at rest, the channel is open. When applying pressure, the membrane gets into the channel, thereby closing the channel. So as I said, it's a relatively old technology. We haven't invented it definitely at Micronit, but it used to be based, at least in the early days, on PDMS because of the need of an elastomeric layer. So what we have done at Micronit, we have developed a portfolio of strategies that would allow us to translate these PDMS-based flow control elements into scalable solution that would allow us to manufacture products that integrate complex flow management and complex workflow. Here I have three examples in this slide. This first example is an early prototype for a product we made for a customer, which consists of 3 layers of COC, 2 thermoplastic, rigid, and 1 elastomeric, in between, all directly bonded. And here we can see the blue layer is the fluidic network, and the white layer is the pneumatic network, which is used to actuate the valves. 3 valves in series, actuated in series, provide flow, so they provide also pumping function. And in this case, we have also microstructured a nozzle at the exit valve, which can be used to dispense very accurate amounts of reagents, for example, to a reaction vessel. These 2 examples here, they are microtiter plates with smart functionalities, with integrated flow control management, which is based again on these valves and pumps. These small round features here and here represents valves, and sets of 3 of them represent pumps, and they are used to drive fluid within this network of channels. And the central portion in this case contains a number of culturing areas which are interfaced with a microelectrode array sensor, which is used for online sensing, and it's mounted on a PCB, printed circuit board. The second type of flow control I want to mention is capillary flow control. In capillary flow control, flow is basically determined by the geometry of the channels and by the surface properties in the channels. So like in this case, you see the blue fluid flows spontaneously inside the channel up to this geometrical restriction and then expansion, which constitutes the valve, basically. These valves, these flow control elements can be fabricated, for example, by hot embossing in polymer, or when scaling up by injection molding. Of course, if we want to perform a multi-step assay, like the workflows that we have seen in previous slides, we also need to add a time component. So once the flow stops at the valve, we should be able, at the right time, to re-trigger the flow. And this can be done by electrostatic triggering. So by patterning thin film of metals, so metal electrode before and after the valve, and when applying a short pulse of voltage across these electrodes, that will trigger the flow to continue. In this way, we can add the time component. As I already mentioned, we fabricate these kind of devices by embossing. So we have an embossed layer, we have a layer which is patterned with electrodes. We bond them together, direct bonding without adhesives, and then after singulation we get this kind of devices. So as you see here, we have a number of incoming channels. Each of these channels displays one of these capillary valves, each of them with their electrodes for actuation, and this can provide sequential flow that can be used, for example, for running an immunoassay, adding sequentially reagents. The third functionality is integration of reagents and surface functionalization. There are different, so in most cases for point of care diagnostic test, integrating reagent is a must. Again, for the sake of automation and ease of use for the user. And so reagents that are more labile need to stay, to retain their activity also after a certain shelf life. So liquid storage is not an option. In that case, we do dry storage of reagents, for which we use different dispensers or spotting machines to be able to dispense the right amount of reagents that are then dried. In other cases, for example, for buffers or less labile reagents, they can also be stored in liquid reservoirs, like in this case, blisters are typically used in point-of-care microfluidics. Surface functionalization is important for a number of reasons. Anti-fouling properties are very important to prevent fouling of the surface or losing the analyte of interest. But also the contact angle needs to be, in some cases, very well controlled. Think of the capillary-based flow elements or devices that we've seen in the previous slide. Here in this movie and in this image here, you see some demonstrators that were developed by Surfix, this one in collaboration with us in the code Pocket project, where you can clearly see the channel with a capillary stop valve and different kind of coating. So a hydrophobic coating is locally used to increase the strength of the valve. A PEG-like coating is used to both control, to make sustain the capillary flow inside the channel and also for anti-fouling surface purposes. And in this movie, you can see different channels which are filled by capillarities, and you can clearly see different flow behavior in different channels that have been patterned with different surface coatings. The last slide on the technology, and then I will conclude with a summary, is about integration of hybrid elements. Many of this imaging has already come forward in previous slides, so I will just very briefly point out or remind you what they are. So this was a work which was done together with NXP and Janssen Diagnostics, which resulted in a publication in which we demonstrated that a CMOS sensor made by NXP, flip chip bonded with a microfluidic device with patterned electrodes that we fabricated, could be used for beads-based ELISA, providing better sensitivity and lower limit of detection than conventional ELISA systems, thereby providing, or ELISA plate readers, thereby showing the potential of microfluidics and microtechnologies. Also, these demonstrators, so we have discussed it already in a previous slide for the sample preparation. For the coil, the planar coil, this planar coil is also a nice example of hybrid integration done at IMEC. And in this case, we have an immunofield effect transistor sensor, which is used for the detection of biomarkers for heart failure from saliva. This is a demonstrator that was made, is being made actually, it's being developed in the CardiaTool project. Again, the plate that we have developed together with IMEC, here you see a multi-layer polymer plate for culturing of cardiomyocytes in a network of channels with integrated flow control, which is interfaced and integrated to a microelectrode array, silicon-based, made at IMEC and mounted or assembled on a PCB. This demonstrator is an electronic nose for early detection of disease developed in the LKOS project. It's a borosilicate microfluidic chip with silicon sensors, which are either chemiresistors based on gold nanoparticles and field effect transistors equipped with silicon nanowires. And the last example is a different kind of component, which is a microlens used in medical imaging. You can see here a part or a close-up of a wafer that would contain 15,000 lens cavities, and it's used for wafer level packaging. This picture here shows a close-up of 4 of these lenses, and in the schematic we can see a glass wafer with a polymeric molded microlens layer on top of it, which should be packaged. When using glass and etching glass, of course, we are limited by the thickness of glass and by the processing parameters. And in this specific case, the depth of these cavities, as well as the residual layer of glass, they are critical. So to overcome these problems, we use a layer of resist, which can be used both for sealing purposes, for the bonding, as well as for as a spacer. This was my last slide, so I will move to the summary now. And I really hope that in this webinar I have been able to show you that in vitro diagnostics is driving medical innovation, enabling personalization and decentralization of healthcare. I also hope that I've been able to show you that microfluidics and MEMS are key enabling technologies and will continue driving innovation and growth in the diagnostic market by enabling integration of functionalities and thereby also automation of diagnostics assays. And finally, I hope that I've also convinced you that with over 20 years of experience in ISO certified manufacturing facilities and with the supporting ecosystems of trusted partner, Micronit is the leading innovator and global partner in design, development and manufacturing of microfluidics and MEMS consumables for the in vitro diagnostic market that can also help you to develop and manufacture your product. I would also like to remind you, please, not to forget that in 1 week time, so next week on Tuesday, June 2nd, my colleague Linnars will tell us all about droplets in a webinar called entitled Let's Make Droplets. And with that, I'm going to move to the Q&A session. We still have a few minutes. Okay. So I see we have a lot of questions, so I'm trying to read. They just came in fresh from the chat. I have here a question that refers to the current pandemic situation. So how can point of care be integrated with data collection such that results from point of care devices can be documented for individual tests during pandemic situation. So if I understand this question correctly, it refers to more data management. Yeah, it's a very good question indeed. Maybe it's good to mention that this is one of the items that came up, or that this COVID situation or pandemic has brought up. It has brought up a number of issues that we didn't even know we had to deal with, or we were taking all our time to deal with. From a regulatory perspective, for a number of test perspective, and indeed, as pointed out by this person, also the data collection. I would say that there is already a lot going on in the field of diagnostics in terms of creating softwares and clouds to be able to collect data and share them, and create apps to be able to connect the collection of data to central databases. I would say that that's also one of the interesting challenges that has been brought up by this COVID-19 pandemic. And yeah, I believe that indeed the use of apps and softwares that can collect information and then share them on a platform with healthcare provider, that would be a way to go. I hope I understood the question. So what are diagnostics applications for hybrid, for example, PDMS and thermoplastics? And what are the major challenges using PDMS? What are diagnostics applications for hybrids? I would say most of them, and the reason is realizing the fact that, as I mentioned, the automation of the workflow, which is required in order to provide simplicity in the use or ease of use of in point of care settings, brings a lot of functionalities together, and each of these functionalities might have requirements that point to a different type of material. So with that respect, I really believe that the integration of hybrid material and hybrid component is needed, and it will be even needed more and more for more applications in diagnostics, in particular at the point of care, because it's really intrinsic in the requirements of this type of tests and field. The question referred particularly to PDMS and thermoplastics. I would say that, so if I read PDMS and thermoplastics, I think of elastomer and rigid substrate. So definitely going back to the active valves, which was one of the functional elements that I have presented. PDMS has been used as a first material, I think, when these type of valves were invented many years ago. We have also made devices also for customers in the years ago based on glass and PDMS. However, the problem with PDMS is that it's not scalable. So especially when you go to diagnostics, when you go to point of care, typically volumes of devices that are required is quite high. So that means that you really need to have scalable solutions that can be automated to manufacture these devices. So with that respect, PDMS is definitely something that we try to go away from, and that's why our efforts have been really focusing on, even for functional elements that, like these active valves where PDMS was used historically, we have tried to come up with alternative materials that would provide the same elastic property to be able to provide the functionality which is needed, but that it's compatible. Well, first of all, that would allow us to do direct bonding with other materials, thermoplastic materials that we make the devices with, but also that would provide compatibility with typical applications in the healthcare area, which is where PDMS is not the material of choice. So I think maybe I will see if there is another question that I can answer quickly, because I'm really running out of time. We have here a question on bonding. How do you bond your substrate, especially when working with heterogeneous materials? So we do have, I haven't elaborated on bonding methods, but clearly indeed when working with different materials, and in particular when combining different materials, it's important to have also a broad choice of technologies for bonding and sealing of microfluidic structures. So over the years we have developed a number of different technologies. In some cases the use of adhesive is needed. So in some cases, for example, for hybrid integration of silicon chips to a polymeric cartridge, then we need to really look into specific material requirements and so on. But in most cases we do direct bonding, which is with some sort of activation. The activation again varies depending on the product requirement, the application requirement. Unfortunately, I am not able to give a lot of details on this because, of course, we have our own secret recipes, but either within Micronit or even looking broader within the Flow Alliance, we can definitely offer a number of different methods for bonding different materials. So I have received really a lot of questions, so there is no way I will be able to answer them today, also because I'm already out of time. But I promise that I will handle all the questions, put them in an email after anonymizing them, and I will share them with you as soon as possible. For now, I would like to thank you for patiently having listened to me, and I will give the word back to my colleague, Jose.
Thank you for visiting our webinar. We hope you liked it. Monica also told it, but we want to invite you for our next webinar on the 2nd of June. Then Eric Linnartz will give a presentation about lead make droplets. In the chat you can find the link to the exit survey. For now, have a nice day.